All right, so let's talk about, I like to recycle, even though I think it's ludicrous that I have to pay money to recycle for them to come pick up my recycling in South Carolina. But I like to recycle, and so does your body. So, all right, your body is actually going to recycle urea, all right, between two regions of the renal nephron there, okay? And predominantly, it goes back and forth between the ascending limb and the collecting duct, okay? There's a region in there in which urea is going to cycle through in that portion there. And we'll see, right, as urea is being removed from the tubular fluid here by the collecting duct, right, it will come from uniporters, which again are another transport protein, all right. At the same time, it's going to enter back into the tubular fluid at the thin segment of the ascending limb, which is right here. Here's the thin segment of the ascending limb, okay. This is the thick segment, this is the thin segment. Not a very big part. All right, but that's where our urea, well, let's see if I have it on here. Whoops, wrong one. Well, you can't see it here, all right, but if we were to continue our drawing, all right, this would be we get to our distal convoluted tubule, and then eventually we'd reach the collecting duct and the collecting tubules here. So it's pretty much, well, let's continue this down. It would be in this region here where we would see a lot of recycling. All right, when it comes to the urea there, okay? But again, this urea cycling is going to help build that concentration gradient. A lot of resorption is dependent in the nephron loop there with establishing a concentration gradient. And urea cycling is one way to help with the establishment and creating that concentration gradient. Because look right here, half of the solutes, all right, are going to be in the interstitial fluid there, all right? And if you recall, solutes are almost like a magnet to water. Water always wants to go to the area, all right, of a higher solute concentration, all right? And it's because that's where there's a lower concentration of water. And so water wants to move down its concentration gradient. So you can look at it two ways. And so this urea cycling helps to set up that concentration gradient, all right, as we get down to the bottom of that nephron loop, all right, we'll see that the interstitial fluid is going to have a significant amount, all right, of solutes there. And you can also see why it promotes, all right, the movement of water, all right, out of the vasa recta, right? Because we have more solutes out here in the interstitial fluid. And so water is going to want to jump out there, all right, because it's going to be moving down its concentration gradient by the process of osmosis. All right, so if we see here, all right, when we're talking about our concentration gradient, we're going to do a quick review here on resorption and secretion, all right? So, a couple points to keep in mind, all right? After we've filtered stuff out of the blood, we are going to see most of those substances get resorbed, all right, in the proximal convoluted tubule. All right, so we're going to see, basically what we're going to see is, all right, most substances have a choice of either being mostly resorbed or mostly secreted. Now, we talked quite a bit about how a lot of those substances are going to be resorbed, and you'll see that. All right, but it's either going to be, it's not a 50-50 thing. It's not like some gets resorbed, some gets secreted. All right, it's either most of it's going to be resorbed or most of it's going to be, excuse me, secreted here. All right, so when we talk about our concentration gradient, we're talking about our nephron loop that includes the vasa recta. All right, we just talked about what goes on in the nephron loop. All right, with our counter current multiplier. all right, and that countercurrent flow there, okay, that's going to occur in the nephron loop. That's going to help to set up our concentration gradient. All right, what occurs in the vasa recta, and finally I just got done talking about the urea cycle. This is going to help us to create that concentration gradient for the interstitial fluid. All right, this is going to help us allow, remember, when you see concentration gradient, you're thinking that's our platform. That's our platform for the movement of substances in and out of things, namely the tubular fluid out into the interstitial fluid and then into the blood vessels, into the capillaries there, okay? So antidiuretic hormone, all right, needs to have a concentration gradient available for it, all right? So it can facilitate the movement of those substances there, okay? Now, when we talk about the regulation, all right, regulation usually occurs in the distal convoluted tubule, the collecting tubules, and the collecting ducts. All right? The cells that are responsible for that are going to be our principal cells and the intercalated cells. All right? So all these things, all right, are governed by a concentration gradient. All right? Movement of things, unless we're dealing with an active transporter there. All right? But when we look at the final product, our urine, all right, we're going to see water, dissolved substances, and waste products. But water and some of those dissolved substances, all right, have been on a long journey. They got filtered out. Most of them got resorbed. But if we find them back in the urine, all right, that means that they were secreted, all right, and so they had to take an extra step, okay? So understand, in a normal situation, as long as there's no pathologies present, all right, if you think about it, substances are either going to be resorbed or secreted, and if something that normally is resorbed, all right, gets secreted, then we have to start asking ourselves, okay, what is causing this? All right, if it's water, all right, normally that's a resorbed substance. Okay, but there has to be something that you call into question in regards to your hydration status, for example, or blood pressure. All right, but eventually with our urine, all right, urine is going to drain out of the papillary duct into the minor calyx, into the major calyx, into the renal pelvis, and then out of the renal pelvis, all right, into our ureters and down into our urinary bladder to be excreted through the remaining portion of the urinary tract. So I want to circle back here and talk to you about our GFR, all right, our glomerular filtration rate, okay? Because again, this is something that is quite important for you to understand, and we use it to help determine, all right, how healthy or unhealthy kidneys are, all right? So as a reminder, glomerular filtration rate is the rate of the filtrate, that's whatever that comes out of the glomerulus, all right? How much and how long, okay? So it's going to be volume in units of time, which is usually 1 minute. So essentially, how much filtrate is made in 1 minute. So in real life, right, if you're going to a nephrologist and they think there's something wrong with their kidneys and they want to measure your glomerular filtration rate, they're going to give you an inulin, not insulin, but inulin injection. All right, it's primarily sugar. All right, and the nice thing about inulin is that it's all filtered. Okay, it's all filtered, so we can tell how much we're putting into your kidney. Okay. All right. So what we'll do is, all right, we'll collect, all right, your urine sample here, all right, and then we'll measure how much urine you're producing, all right, and then we'll check out what's in your urine. Okay, and when we do the inulin injection, we do it over a period of time. Okay, there's going to be several different collection times, and then we're able to calculate and figure out, all right, what your glomerular filtration rate is. So the equation here, glomerular filtration rate, is going to be the concentration of inulin in your urine, so what you're peeing out, all right, over, all right, well, that multiply it by how much you're producing, all right, and then it's going to be divided by the value of inulin in the plasma. So essentially, when you receive the injection, we're going to compare how much inulin is actually in your blood compared to how much you've peed out over a specific period of time, all right? And that's how we'll be able to calculate the GFR. Normally, 25 milliliters per minute, okay? If it's lower than that, then we're going to be suspecting there's something going on with the kidney. All right, and there could be several reasons for that. Decreased GFR could be decreased blood pressure. All right, there could be several reasons going on for that. All right, another way to check how well the kidneys are functioning, we can do a renal plasma clearance test. All right, and so basically, all right, what we can do is give you a specific substance and see how long it takes for that to come out. All right, we can put something specific, another injection into the plasma, and we can do the same thing. We'll give it over a specific period of time. We'll see how much we collect through the urine, all right, and then we can determine whether or not, all right, what the glomerular filtration rate is, depending on how long and how much of the substance we see, okay? All right, this is a great value. I know I keep going to this, to the creatinine clearance, all right? But basically, if you measure the creatinine, because it's only going to be a little bit higher than the GFR, so to determine, if you're able to determine the creatinine clearance, all right, this is an easy way to figure out what your GFR is going to be. All right, questions about that? No. All right. So let's talk a little bit about pee. All right. When we discuss what urine is, yes, it is a metabolic waste, all right, substance here. All right. But basically, we can assume that anything that we find in the urine, all right, was filtered, all right, and then it was possibly resorbed and secreted, okay? So anything that was once in your blood that got filtered out, all right? And so it gives us a pretty good idea of what's going on with the blood plasma. Now, I don't really stand by this, but I think I've mentioned it to you before. I mean, we do consider urine to be sterile, all right, unless there is something in the urine that would not make it sterile. Like if you had cloudy urine, usually that's an indication of white blood cell elevation and a good possibility that you might have an infection of some sort. So obviously someone with a UTI would not have sterile urine, all right? But it is considered to be a relatively sterile substance here. So when we discuss the characteristics of urine, all right, we are going to discuss what's in it, all right, how much, all the time. If you go to see a urologist, they will ask you what you think or how much urine you're producing, especially if you're describing any issues that when it's related to your urinary tract, all right. They're going to ask you, is there any smell to it? What does it look like? Is it cloudy? Is it clear? Is it yellow? Is it dark yellow? Is it brown? Is there a red tint to it? All right, so we're going to talk about the color of it. Is it clear or is it cloudy? And then we'll check to see how much water is in it, and we'll talk about the specific gravity value. And then, is it acidic or is it alkaline? Okay, meaning, is there a lot of hydrogen ions in your urine, or is there very little hydrogen ions in your urine? Okay, so the majority of your urine is going to be made up of water. Okay, 95% is water. The other 5% are going to be solutes. A significant portion of those solutes are going to be waste products. If you're taking any drugs, all right, then we'll find some of the remnants of the drugs there, and even ketone bodies will be present, which is part of our protein metabolism here. All right. I find urine dipstick tests to be highly beneficial in determining some of the health status of my patients, because it can tell you quite a bit on what type of diet they have, all right, and kind of gives you an idea if they are depleted in certain substances or if they have too much of a specific type of substance, if they have an excess of something. So when we talk about urine, let's talk about the characteristic of volume. All right, 1 to 2 liters a day, depends upon, you know, how hydrated you are. All right, the amount of fluid intake that you're taking. Folks with higher blood pressure, all right, will produce more urine. Folks with lower blood pressure will produce less urine. All right, if you're taking any drugs, like I was telling you about spironolactone, which is a diuretic. That's a drug that we give to folks with high blood pressure when we're trying to increase their urine output. All right. Also, folks with diabetes. That is one of the symptoms of diabetes is increased thirst and also increased urination. Okay. So you can see, right, you need to have at least half of a liter amount of urine, all right, to produce, so you can get rid of the metabolic waste substances of your body, like nitrogenous waste and whatnot. Okay. So if you fall below the value of 0.4 liters, then we start to get really concerned, and you can start to have an increase in metabolic waste products in your blood. And usually when that happens, you're going to affect the pH value of your blood. And when you affect the pH value of your blood, and especially if it moves it into the lower pH, then you're going to cause denaturation of proteins. Your proteins will start to break apart, and that's always a bad thing. Okay. Speaking of pH, right? The pH value for your urine can go from a relatively high, or excuse me, low pH, which is particularly acidic at 4.5, to a slightly basic 8.0. Okay. So certain things will increase the acidity of your urine. All right. Protein plays a big role. Folks that take that supplemental protein, like whey protein, soy protein, they have plant-based proteins now. If you do supplementals, anyone ever tried a plant-based protein shake? I tried it for the first time today. It was god awful. All right, but I'm going to stick it out because I do think it's going to be better in the long run. All right, now we're talking about less acidic. That's going to be folks that are going to have diets high in fruits and vegetables here. All right, so the pH is there's a lot of variables that are going to affect it. Obviously, if you're high metabolism, all right, and you are undergoing a lot of chemical processes in the body, some of those waste products will yield substances that will have a more acidic nature. All right, so of course, you know, when the blood becomes acidic, all right, when the pH starts to drop in the blood, we are going to excrete those hydrogen ions into our urine. Therefore, our pH value will start to decrease. Okay. All right, specific gravity, think of water. All right, and that's basically what we do when we look at the specific gravity. I have done urine samples for DOTs, and people, you would have thought that they actually just poured tap water into the urine specimen cup. All right. Because it looks so clear and water-like, all right? But it just means that they're very well hydrated, and their specific gravity is that of 1. Basically, we're saying that that urine sample, all right, is pretty much the same density as water. It can only increase, so your specific gravity can only go up. And what causes it to increase is the presence of solutes in that urine sample there. Okay, color, like I said, we can go from clear to dark yellow. If it's dark yellow, we're going to start to suspect an increase in concentration of the urobilin, which is one of the intermediate breakdown, well, not an intermediate, this is one of the waste products of hemoglobin. Okay, So if you see an increase in urobilin, then you might start to suspect, okay, this person is actually having an increase in red blood cell breakdown. That's the problem. All right. It could be several reasons. So there could be some sort of anemia that's suspected. Okay. So if we increase our urine, all right, then we'll see a lighter color in that. Or how about the smell? All right. This is an interesting term here, urinoid. All right. That's a specific word to describe the smell of pee, all right, of your urine. So urine should not have a fresh smell to it. No, that's the word I'm using, not fresh smell. That's a horrible description. Sweet smell, excuse me, a sweet smell. When it's a sweet smell, usually it's the presence of ketone bodies, and that's usually a byproduct, all right, when somebody is undergoing the metabolism of proteins more so, but diabetics most commonly will have, all right, a sweeter smell of urine. Part of that is because they'll have more glucose getting spilled into their urine, right? So they describe it as a fruity smell, right? But you definitely don't want your urine to have a sweet smell to it or a fruity smell, okay? Normally it has that, I don't know how else to describe it, the urinoid smell. We've all smelled our own pee, so we all know that. But if you've ever been to Hawaii or an island, has anyone ever heard the saying, if it's yellow, let it mellow. If it's brown, flush it down? Has anyone ever heard that? No? All right. On certain islands, because fresh water is, you don't want to be wasting it. So the policy is on some places that if you're just going to pee, don't flush it. All right. Don't flush it. If you're going to defecate, then obviously flush it. But if the urine sits over time, the ammonia smell starts to break down and it will reek. And I went to somebody's house who hadn't flushed their toilet in over a week, and I walked into, again, I walked in there and it smelled like somebody had just cleaned the whole bathroom with ammonia, and it was just from that toilet. So I definitely flushed it. All right, I know that probably ruined them, but all right, mixturation. Let's finish up and talk about how we make pee. Now we're going to talk about how we get rid of it. All right, and there's two reflexes that are going to be involved: the storage reflex and the mixturation reflex. This is going to involve, all right, the autonomic nervous system, the sympathetic division, the autonomic division. There's also, though, when we go to the bathroom, when we pee, there's the somatic nervous system. That's the part of the nervous system that we can consciously control. We'll talk about that here in a second. All right, but when we talk about the reflexes, we're just talking about the autonomic nervous system only, okay? All right, so let's talk about, all right, what part of the nervous system controls what. All right, so when we're talking about mixturation, the act of peeing, we're going to be talking about the urinary bladder and then the urethral sphincters. Remember, there's two. There's an internal urethral sphincter and an external urethral sphincter. All right, so when we're talking about the urinary bladder, you have sympathetic, parasympathetic, and somatic innervation, which are going to help with expelling urine from your body. So let's start off with the sympathetic one. You need to know this, T11 through L2, all right? Those are going to be the spinal nerve levels that are going to cause, this is when we're discussing mixturation now, the contraction of the internal urethral sphincter, all right? And what it will also do is it will inhibit the contracture of the detrusor muscle, all right? What's the detrusor muscle? That's the muscle that sits in the wall of your urinary bladder, all right? So when it's time for you to use the lavatory, all right, that muscle will contract, it'll squeeze the wall of the urinary bladder, and it'll push the urine out, all right? But that urine will not leave if the internal urethral sphincter is contracted. Okay, we have to relax that. We're going to talk about that here in a second, though. I just want to go over some of the components here. All right, the parasympathetic, all right, it's the origin, all right, the cell bodies are going to be located in the pons here, okay? And we're going to utilize the pelvic splanchnic nerves. And those nerve roots come from S2 to S4, all All right, that's the distal or the caudal portion of the cranial sacral or the parasympathetic division here. Now, when the parasympathetic division is active and stimulated, it will contract the detrusor. All right, it will squeeze the walls of the urinary bladder and it will cause relaxation of the internal urethral sphincter. So that will allow the movement of urine out of your bladder or mixturation. All right. Then the final component from the somatic nervous system comes from the pudendal nerve. All right. This is the nerve that innervates the muscles that you learned how to use when you were potty training as a little one. All right. That innervates the external urethral sphincter. Okay. And so normally, all right, this contracts the external urethral sphincter, and that will prevent urination until you want to relax it, and then you can allow the flow of urine. All right, so let's break it down real quick here. We're almost done. All right, remember what I said when we talk about mixturation. There's two reflexes. All right, there's the storage reflex, and there's the actual mixturation reflex. The storage reflex is constantly going on. All right, you know how we talked about the vasomotor tone, all right, for your blood vessels, how the sympathetic nervous system is always releasing neurotransmitter from the sympathetic nerves onto the blood vessels there to keep them partially constricted. That's happening all the time. All right. Well, for the storage reflex, the sympathetic nervous system is doing the same thing. It's going to constantly, all right, release, all right, its neurotransmitter that is going to cause relaxation of the detrusor muscle. Okay? When it causes relaxation of the detrusor muscle, it allows the urinary bladder to fill with urine. At the same time, it causes contraction of the internal urethral sphincter so we can keep the urine in the bladder. Okay? Meanwhile, the external urethral sphincter is constantly being innervated by the pudendal nerve. So it too is contracting those muscles. Again, to retain urine, keep it inside the bladder. Okay? So this is, you're not even thinking about your pudendal nerve right now. All right? But it is stimulating the external urethral sphincter here. So if you look here on our picture, all right, when we're talking about the storage reflex here, all right, we're talking about T11 through L2 spinal nerves are going to contribute to causing relaxation of the detrusor muscle here in the urinary bladder, all right, but at the same time, it's causing contraction here of the internal urethral sphincter. So as urine builds up here, we're good to go, all right? Meanwhile, the external urethral sphincter, all right, down here in the pelvic floor, all right, levator ani muscle and group, all right, they're going to be undergoing, all right, innervation from the pudendal nerve to sustain a contraction here. So no urine is leaving, okay? That's the storage reflex. That's going on right now in all of us, okay? I have to assume so. Otherwise, you'd be making a beeline to the bathroom, okay? So next is the mixturation reflex. You can't read that, okay? So now our bladder is starting to fill up, thanks in part to the storage reflex. So we get up to about 200 to 300 milliliters, just to let you know, a can of Coke is 355 milliliters. Okay, so that's the volume that we're looking at to start to get that feeling of like, hey, I might have to pee. Okay, because as we start to fill the bladder, we're going to stretch the walls of the bladder, the baroreceptors in the wall will be stimulated. And now we're going to start, all right, to undergo the reflex of mixturation here, okay? So those baroreceptors send that sensory information, okay, up to the pons in the brainstem there, okay? And then the pons then is going to send that information back down the spinal cord, all right, to the pelvic splanchnic nerves, S2 through S4, all right? And it is going to now tell, all right, pelvic sphincter nerves will now tell the detrusor muscles to contract, squeeze the walls of the bladder, while at the same time, it's going to cause the internal urethral sphincter to relax. So urine can start to come out of the bladder. But we're missing one other thing. We've got the external urethral sphincter. That is under, all right, the pudendal nerves here. Okay, the external urethral sphincter, the pudendal nerve, we need to relax that. All right, when we do that, then you're able to, you can actually go to the bathroom and pee. So that's what this picture here is showing us. Here's the full, not full bladder, but the relatively high-filled bladder, stretches the urinary bladder walls, sends that information up to the pons, all right, via the baroreceptor sensory neurons. The pons then stimulates, all right, the pelvic sphincter nerves to contract the walls of the urinary bladder. The detrusor muscle here starts to contract. At the same time, it relaxes the internal urethral sphincter, all right, and then we relax the pudendal nerve and urine leaves out through the urethra there. All right, so this comes down to the conscious control of urine, all right, when we're dealing with the pudendal nerve. This is when, all right, in the cerebral cortex, specifically in the precentral gyrus there, all right, that's that one area in the frontal lobe, all right, in which we will now inhibit the pudendal nerve, causing relaxation of the external urethral sphincter. And then if you really want to, if you're in a hurry, we've all done it, where we just want to hurry up and pee and get out of there. So if you kind of take a deep breath and then kind of bear down, increase your intra-abdominal pressure. It's called the Valsalva maneuver, all right? And then you can actually contract your expiratory muscles. You can actually get pee moving a little bit faster here. So when you're all done, the bladder's empty. So now the detrusor muscle can relax, all right? And now what we'll do is we will now engage back into the storage reflex, all right? So when that occurs, we'll activate all the neurons of the storage reflex. That's going to be the sympathetic neurons, all right, that are going to cause this, the internal urethral sphincter to contract, all right, and then we'll have relaxation of the detrusor muscle, okay? So last slide, all right there, okay? So this is kind of like what I was talking about when we were dealing with trying to quicken our pee time or whatever. If we're trying to urinate and it's not really working, all right, this is when we can actually call into place the somatic urine or the somatic nervous system there and start to increase that pressure, the intra-abdominal pressure there, okay? Any questions about any of that? All right, well, that's the end of lecture, chapter 24, okay? I'm going to go right into chapter 28 for the lab. So if you want to kind of switch notes, we're going to climb back into the reproductive system here.